So the genetic map of the A plasmids are, it can have a separately here. The genetic map of the F plasmids, but genetic map of F1 plasmids, here is a TRA regions. But here is also TN genes or IS3 genes. Several number of genes are there here. But this bacteria is a spherically can formation of the replication. First part is replication part. The replicated part is attached here. Attachment. So this is also in the cell to outside of the cell. This is also plasmid characters, plasmid structure. Then the second also got a first one is that euplasmid. Then second one or plasmid. Or means also called restensive plasmids. The restensive plasmids are most widespread and well-studied group of the plasmids conforming resistance, hence called the resistant plasmids to antibiotics and virus, other growth inhibitors, antibacteria and virus. It is also separate parts which can be having that plasmid is called a or plasmid. Or plasmid typically have genes that code of the enzymes able to destroy and modify antibiotics. Antibiotics means antibiotic quality. It is important for the character features of the organisms. So this is also important. Then they are not usually integrated into the host chromosome. Some R plasmid passes only a single resistant genes, where other can have as many as 8. Many of the 8. So plasmids are means resistant plasmids are 100, for example, is a 94.3 kilobases pair plasmids. Figure 5.3 next to this one is also 5.32. Then is also genetic map of the resistant plasmids, the replication. So which is also then 94.30 here 25 is this all is also gene located in a circular type of the manner that is called a R plasmids. Then R plasmid is also simple. Only resistant qualities are located on the spiral or spherical peripheral part of the plasmids. That is called a R plasmids. But here is a virulency plasmid. Virulency means it is also virus, means also poison. It can be that virulency plasmids. It can killing the other type of the bacteria, also got other type of the virus. Virulency plasmids confer pathogenicity on the host bacteria. They make the bacteria more pathogenic as the bacterium is better able to resist host defense to produce a toxins. The toxin substance are releasing of the plasmid is called a virulency plasmids or a VIR plasmids. For example, plasmid, tumor-induced plasmid of agrobacteria to induce crown gall disease of angiospermic plants, which is the best type of the plasmid to be used to gene cloning or agrobacteria gene transformation. The gene transformation, this TI plasmid is separate or tumor-induced plasmids. The tumor is a formation. The tumor is which can transfer of the one cell to the other cell of the genes. That gene is present in this bacteria. This bacteria is which can use it to the gene transformation. That is a one type agrobacteria mediated gene transformation. Then enterotoxinic strains of a Escherichia coli bacteria causes traveler diarrhea because of a plasmid that codes for an intraxin which induced the extensive secretion of water and salts of into the bowl. This is also secretion qualities are also present in the virulency character of the plasmids. Then also base means what are the virulency means that plasmids may be release of the toxic substance. It is also examples of the agrobacteria mediated gene agrobacteria tumescency, agrobacteria rhizogenesis. These two types of bacteria which can causes of the release of toxin substances to the other cells. That cells are killed. This is also then the fourth one coal plasmids. The coal plasmids are carry genes, so that is also that genes are that confer the ability to the host bacteria to kill other bacteria by secreting bacteriocins. So this bacteriocins are separate. Then example is a bacteriophage like then a type of the protein bacteriocins often kill cells by creating channels in the plasma membranes. Through the plasma membranes, that the plasmids are that enzymes which can kill the other cells, thus increasing its permeability. They also may be degrade DNA or RNA or attack so peptidoglycan and weaken the cell wall. Weaken the cell wall under the through the plasma membrane channels. The whole plasmids are enter to the other bacteria cell, other plant cell. That plant cells are killed. That is so bacteriocins acts only against closely related strain called E1 plasmid or E. coli code for the synthesis of a bacteriocins called a colicins, which kill other susceptible strains of E. coli bacteria. Coli plasmids of some E. coli code for synthesis of bacteriocins, namely calcicin, that kill enterobacteria species. Enterobacteria species are killed the poison substances. That poison substances are releasing plasmid is a cool plasmids. Lactic acid bacteria produce the bacteriocin, nisin, which strongly inhibits the growth of wide variety of the gram-positive bacteria and used as a preservative and food industries. So this is also preservative in the industries, storage, food materials, storage parts. It can affect to the causes of the toxic substance are releases. That food materials are also damaged, also good. Then also fifth one, metabolic plasmids. The metabolic plasmids also called the degradative plasmids. Degradative, it can pass as genes to code enzymes that degrade usable or unusual substances such as toluene, aromatic components, also pesticides, 2,4-dichlorophenoxy acetic acid, and sugars. This metabolic means degradable. Degradable means degraded, the degraded food materials. Also got TOL, plasmid of a Pseudomonas putida. It is an example. However, some metabolic plasmids occurs in certain strains of rhizobium, induce the nodules formation. In the rhizobium nodules, the metabolic means metabolities or metabolic activities, which releases that bacteria specialized cells are formation, specialized nodes are formation. That is got root nodules. These root nodules inside of the root nodules, which are in the rhizobium bacteria are present. That is examples of leguminaceae family members. It is also major important of nitrogen fixation in the symbiotic relationships. The rhizobium induced nodules formation in leguminaceae and carry out fixation of atmospheric nitrogen. A brief summary of important types of bacterial plasmids, their host and properties are given. So what are the properties? What are the given properties are here is also, this is also. Then the host qualities are there. But this is also got a list of the plasmid, type of the plasmids, purpose and examples. So this is also the cloning vectors. Then DNA cloning and replications. PBR 322 use the expression vectors. Then protein productions. But the it is a summarized to the short to easily remembering that topic of type of the plasmids. What are the purpose? What are the examples of the type of the plasmids? Then first one is a cloning vector. That is also cloning. Cloning means copies or multiplication of the DNA in the vector when inserted to the vector to be cloned. That is the second point is also expression vectors, the protein productions. That is examples of PGT and PGEX, reporter vectors. with the gene regulating studies, gene regulating, gene expression, gene cloning, and also gene therapy. That is also last characters are there. Then also called as shuttle vectors. So used in a multiple organisms by AP and PSV2. Then RNA gene knockdown type of the plasmid. It is also gene silencing. Then viral vectors, high efficiency of gene delivery. That is also one purpose of the separate of the and plasmids are there. But get a transpose-based vectors. Gene insertion, random specific. Also examples of PMODR. So gateway vectors. These are also several types. Out of we studies only 5 types are the cloning vectors, expression vector, reporter vector, shuttle vector, gene knocking vector, and viral vectors. That is also based on the functioning. Also functionings are there, but that is also the next part. Here is the types of the artificial plasmids. These are all is also natural plasmids, which can classify this one only 5 type of the plasmids based on the only 3. But it is a plasmids, R plasmid, cool plasmids. But now, which is also a plasmid is also over. Then R plasmids. Then here is also virulency plasmids. But here is a cool plasmids. But here is a metabolic 5 types. The 5 type of the plasmids. That what are the function of the 5 type of the plasmids. This is also types of artificial plasmids. The artificial plasmids are engineered DNA molecules used as vectors to introduce the replication. For gene replication, our biotechnologist, biochemist who has prepared the artificial DNA, that is artificial plasmids of DNA, that is called artificial plasmids. They are specially designed for specific application, such as cloning, expression, genes. 3 types. The 3 characters are based. Our artificial gene cloning is gene or DNA, also called plasmids, are reported based on the expression, cloning, and gene silencing process. First one is also cloning vector purpose. The cloning and replicate